Duplicated genes and pseudogenes. Duplicated genes. They usually encode closely related proteins. They are clustered together in the genome. They are formed by duplication of an ancestral gene followed by a mutation. What are pseudogenes? Non-functional copies of the genes. They are also formed by duplication of ancestral gene, or it can reintegrate by the process of reverse transcription into the genome. This is the example of duplicated genes. Here we can see that there are 5 functional genes and there are 2 pseudogenes. How they produced? This is epsilon. These two, they are gamma. This is gene delta. This is gene beta. So these are 5 genes which are functional. While in addition to these 2 genes, we see that there are 2 other genes, this one and this one. So beta 1 and beta 2, they are pseudogenes. So this is that how the genes are duplicated and what are the functional genes and pseudogenes. So in addition to these 5 functional genes which are epsilon, gamma, delta and beta, we can also see that on chromosome 11 on the human genome there are 2 other genes, beta 1 and this beta 2, they are pseudogenes. What are pseudogenes? Pseudogenes usually not expressed due to the mutations which are present in the sequence of the genes. Why they are not expressed? Because due to the particular mutation or a specific mutation that can create a stop codon in the sequence of the gene, or that mutation can prevent messenger RNA processing, or that mutation can prevent the expression of that gene due to the lack of regulatory sequences. So pseudogenes, although that is a sequence of the gene, but those genes, they are not expressed. The expression cannot be due to either the mutation can cause a stop codon, or the mutation can prevent the expression of the messenger RNA, or there can be lacking of the regulatory sequences which are usually present with the genes. So here there are 2 pseudogenes. We can see that 5 are functional genes and there are 2, these are pseudogenes. What is gene duplication? And the example of gene duplication or the duplicated genes is the globin family in case of the humans. Ancestral globin gene was duplicated approximately 500 million years ago. Mutation in both genes to differentiate them as alpha and beta present in all higher vertebrates. The ancestral globin gene was present in the ancestral chromosome or the ancestral genome, and it was duplicated approximately 500 million years ago, and it was differentiated into alpha and beta. Further gene duplications produced alternative forms in mammal as well as in primates. This is the example of gene duplication. The example is globin family in case of humans. Globin gene was initially present in the ancestral genome approximately 700 years ago. This gene was converted into alpha and beta. So beta was present in almost all mammals, and then this beta was converted into adult beta and fetal beta. So fetal beta later on, approximately 100 million years ago, was converted into epsilon and gamma. So gamma has 2 copies, gamma A and gamma G. On the other hand, adult beta was duplicated into two genes, which is called as delta and beta. So this is that how a single gene which was present on the ancestral genome 500 million years ago was converted into 5 different copies of the gene. This cluster are the duplicated genes are present on chromosome 11. Gene duplication. There are two common phenomena which produce DNA duplication or the gene duplication. The first phenomena is called as slip-strand mispriming, and the second phenomena is called as unequal crossover during the recombinations. This is the first phenomena that how DNA duplications occur. The first phenomena is slip-strand mispriming. This is the normal replication process in which we can see two strands 5 to 3 and 3 to 5, and this is the normal replication. But in case of backward slippage, we can see that a small sequence which was denoted as 2, this sequence was backward slipped. So as a result of this slippage, we can see that now the sequence is 1, 2 and 2 and 3. So in the original sequence, it was CAG, CAG, CAG mean box number 1, box number 2 and box number 3. But here we can see that as a result of backward slippage, which causes actually insertion of the specific sequence or a small sequence, we can see that this sequence which was marked as 2 is duplicated. So this is a backward slippage that causes DNA duplication, while the second phenomena is called as forward slippage, which causes deletion. Here we can see as a result of forward slippage, we can see that the sequence of the DNA has been deleted between 3 and 5. Here it was 4. In the original sequence 1, 2, 3 and 4 and 5. Here we can see that 1, 2, 3 and 5. So the 4 CAG, this sequence CAG due to forward slippage that causes the deletion has been deleted from the original sequence. So DNA duplication can be due to the slip-strand mispriming. Slip-strand mispriming can be backward and it can be forward. In case of backward, it can causes the insertion of specific sequence. In case of forward slippage, it causes the deletion. DNA duplication due to unequal crossover. These are two genes, gene 1, gene 2, which are present on homologous chromosomes, and due to misalignment between these two genes, we can see that in the DNA or in the chromosome, both genes, they have been inserted on the same chromosome. So this is the second phenomena of DNA duplication, that if there is unequal crossover, that can also result in the form of DNA duplications. How the pseudogenes they are produced? Pseudogenes, there is a gene on the chromosome or there is a DNA sequence which is functional. During the process of transcription or splicing RNA, this RNA is with the help of the process reverse transcription is converted into the DNA, and this DNA reintegrate into the original sequence of the genome. So with the help of this process, the genes which are functional genes, they are converted into the pseudogenes. The actual gene was present here. We can see that there is a functional gene. This is actually this one, and with the help of this process where the DNA is converted into RNA, and with the help of RNA transcription, again DNA is formed, and that DNA reintegrate into the genome, and this reintegrated genome or the gene is the pseudogene, mean that do not produce any RNA or that do not produce any protein. And other phenomena that how pseudogenes are produced, this is the DNA sequence, DNA, and this is the promoter region, exon 1, intron, exon 2, then intron, then exon 3. So, as a result of transcription, primary transcript mean messenger RNA is produced. Then this messenger RNA, as a result of reverse transcription, is converted into cDNA. This is a cDNA, and this cDNA reintegrate into the genome. So here we can see that exon 1, exon 2 and exon 3, this works as a pseudogene. The actual gene is converted into the pseudogene. So these are two different phenomena by which the duplication occurred. Functional gene, the process of evolution produced truncated gene and gene fragments. Through the process of evolution or through the evolutionary history, many functional genes, they were either converted into truncated genes or they were converted into small gene fragments. So most of these truncated genes or gene fragments, they are non-functional. So if we conclude that what are duplicated genes and pseudogenes, duplicated genes and pseudogenes, they are clustered together in the genome. They are formed either the duplicated genes or the pseudogenes. They are formed by the duplication of the ancestral gene or the ancestral genome. Most of the pseudogene and duplicated genes, they have non-functional copies. Although in case of duplicated genes, there are some of the copies that are functional, but there can be some copies of the gene which are non-functional.